In this video, we will discuss about RNA, ribonucleic acid. Now, what is RNA? What are its different types and what are their functions? Now, what is ribonucleic acid? This is made up of nucleotides. Nucleotides are building blocks of RNA. Each nucleotide is made up of a sugar because this is 5 carbon in length, so this is called as pentose sugar. This is also ribose sugar. This is second component is nitrogenous bases. Now purines and pyrimidines, phosphoric acid, and these nucleotides they will combine to form a polynucleotide chain. You can see this is the single stranded RNA. This is not double stranded, so this is made up of a single strand. Now, so what is RNA? This is, we can see, this is a polyribonucleotide chain because this is made up of many nucleotides. Now, this is single stranded. Now, this is made up of purines and pyrimidines. Now, these are the bases cytosine, guanine, adenine, and uracil. Now, in case of RNA, thymine is not there. Thymine is replaced by uracil. And this has a complementary sequence to that of the DNA template. The sequence on the DNA template is ATACTG and RNA will be UAUGAC. We can see here this is the single stranded ribonucleic acid having bases. So types of RNA, there are 5 types of RNA: ribosomal RNA, messenger RNA, transfer RNA, genetic RNA and catalytic RNA. One by one we will discuss all the RNAs. First we will discuss about ribosomal RNA. To know about the ribosomal RNA, we should know certain terms. This is single stranded, but when it shows folding like this, so this area is called as hairpin loop like structure. This is single stranded, but here this has become double stranded, and where it become double stranded, this show hydrogen bonding between bases of or nucleotides of the RNA. Now total RNA is present in a cell. Out of that, ribosomal RNA is 70 to 80%. So this is the abundant RNA present in a cell. Second term, we should know what is sedimentation coefficient. When centrifugation of RNA is done, how does it sediments during centrifugation? This is called as sedimentation coefficient. Now, if we see ribosomal RNA, this helps in formation of ribosomes. Now, this is the smaller subunit of ribosome. This is the bigger subunit of ribosomes. They lie separate in the cytoplasm, but during protein synthesis, they will combine with each other and they will associate with each other like this. If we see about prokaryotes and eukaryotes, in case of prokaryotes, smaller subunit is made up of 30S. Bigger subunit is made up of 50S, and together when they come associated, this is called as 70S. And in case of eukaryotes, smaller subunit is 40S, bigger subunit is 60S, and when they combine together, this is called as 80S. Now we'll discuss about ribosomal RNA. So ribosomal RNA helps in formation of ribosome. This also helps in formation of the protein. Now this is single stranded, but at certain places this may become double stranded. So depending upon its sedimentation coefficient, RNAs of eukaryotes are of 4 types: 28S, 18S, 5.8S and 5S. And RNAs of prokaryotes are of 3 types: 23S, 16S and 5S. In eukaryotes, 28S, 5.8S and 5S, they occur in the larger subunit of the ribosomes, like we have seen in the earlier slide about larger and smaller subunits, and 18S occur in the smaller subunit of the ribosome. In prokaryotes, 23S and 5S, they occur in the larger subunit of the ribosome, and 16S occur in the smaller subunit of the ribosome. So we can see here, this is the smaller subunit, this is the bigger subunit, like we already discussed. It is, if we see together, so this is 80S in case of eukaryotes and 70S in case of prokaryotes. Now, what are the functions of ribosomal RNA? One is this helps in formation of ribosomes, and this ribosomal RNA also helps in protein synthesis. Now, if we see the details here, we can see here, this is the smaller subunit of the ribosomes, and this is the messenger RNA binding to this smaller subunit. Then bigger subunit will also come and join, and this is the transfer RNA. So ribosomal RNA plays a very important role in the formation of the protein. So this helps in formation of the ribosomes. This also helps in protein synthesis. Second category of messenger RNA. Now, as the name indicating, this carries the messages from the DNA now in the form of codon for the formation of the amino acids. Now, 2 to 5% of total RNA is made up of messenger RNA. Its sedimentation coefficient is 6 to 30s. Now, this become messages from the DNA. Now, codon present on the DNA, opposite to that, complementary sequence will be present in the RNA. So this help in formation of the protein or polypeptide chain over the ribosome. So if we see the structure of the messenger RNA, now this will have, we can see this part is cap. This is the non-coding region 1, this is non-coding region 2, and this is the tail part, and this part is initiating codon, this is the termination codon, and this is the coding region. So this coding region is responsible for the formation of the polypeptide chain. So whatever information is present in this coding region, that will be transferred to the polypeptide chain. If you want to see the detailed structure of the messenger RNA and nucleotides, link is given in the description box. You can see one video of nucleotide, second video of messenger RNA for its detailed structure. Now, next is third type of RNA. This is called as transfer RNA. This is also called as soluble RNA. This is the smallest RNA with only 70 to 95 nucleotides, and its sedimentation coefficient is 4s. To explain the structure of transfer RNA, there are two models. One is clover leaf model, second is L-shaped model. This is a two-dimensional model and this is a three-dimensional model. Now, to study the structure of transfer RNAs, we should know certain terms. Like there are 100 types of transfer RNAs. Sedimentation coefficient is 4s. Clover leaf model was given by Hawley in 1964. So first he gave the structure of transfer RNA which can carry the amino acid alanine. So this is called as alanine transfer RNA. Second was given by Clark. This is called as L-shaped model and this is three-dimensional. Hawley, for giving clover leaf model, also got Nobel Prize for the same. Now there are certain terms like this I sign is called as pseudouridine. DHU mean dihydrouridine. I mean inosine. So first we will study clover leaf model. Now this is the clover leaf model given by Hawley. Now we can see here parts of the transfer RNA. Now this part, this area, this is the 3' end, this is the 5' end. Now we can see this is RNA which is being folded like this manner. So this is becoming double helical at certain places like this. So this is the 5' end. This part at the 3' end is called as CCA part. This side will accept the amino acid or will carry the amino acid. So this is called as acceptor side. This is T-pseudouridine C loop. This loop will help in binding on the ribosome. So this is also called as ribosome binding loop. This is dihydrouridine loop. This will help in binding to the enzyme aminoacyl synthetase enzyme. And this enzyme plays a very important role during the protein synthesis. And this part is the anticodon part which will attach to the codon present on the messenger RNA. And this is the variable loop. Its function is not very clear. This may be giving support to the structure. Now this T-pseudouridine C loop here, this is showing unpaired bases. So 7 unpaired bases. DHU loop will have 8 to 12 unpaired bases. So these are two helical structure which are at right angle to each other. So this is the clover leaf model. Now we'll discuss about Clark's L-shaped model. We can see here this is showing L-shaped structure. This is anticodon loop, anticodon up. This is T-pseudouridine C loop, and this is the D loop, DHE loop. This is the amino acid accepting site. So this is Clark's L-shaped model, and that was clover leaf model. So what are the functions of transfer RNA? Now they, as the name indicating, they are helping in transferring of amino acid to the ribosomes for the protein synthesis, also called as adapter RNA. For each amino acid, there may be different transfer RNA, but sometimes same transfer RNA can carry the same amino acid. So this is called as isoaccepting transfer RNA. Now there are two sites on the ribosomes. This is the A site, this is the P site, amino cell acceptor site, this is peptidal transfer donor site, or A site is also called as adjacent binding site, P site is also called as primary binding site, E is the exit site. So we can also see a figure. This is the smaller subunit of the ribosome. This is the bigger subunit of the ribosome. This is the messenger RNA. This is the transfer RNA. This is the codon part. This is anticodon part. And this is formation of the polypeptide chain. This is outgoing empty transfer RNA. And this is incoming transfer RNA bringing the amino acid. So transfer RNA helps in transferring the amino acid for the formation of the polypeptide chain. This also helps in binding to the ribosomal RNA or ribosome. This also pairs with the codon present on the messenger RNA. Now another kind of RNA is genetic RNA. Now this is the RNA which is acting as a genetic material. I mean, if DNA is not present in an organism, there may be genetic RNA. So this will act as a genomic RNA. This is present in the nucleus. This can be single or this can be double stranded. Now in case of tobacco mosaic virus, this is single stranded, but in case of reoviruses, this is double stranded. Another kind of RNA is catalytic RNA. This acts as biocatalyst. This will play enzymatic activity. This is also called as a ribozyme. So this will also control various biological processes. Now this will also help in protein synthesis. Now catalytic RNAs can be of various types. There are three types. Small nuclear RNAs, snRNAs. They are present in the nucleus. They will complex with the protein to form riboprotein. Now they help in processing of the gene because if we see the RNA, this is made up of exon and introns, and introns they are removed. This removal of intron is called as splicing, and then exons will join to form a process gene. So in that, small nuclear RNA will help. Second kind of catalytic RNA is small nucleolar RNAs. This is found in the nucleolus. This can complex with protein fibrillin. Now, what are its functions? This can convert pre-ribosomal RNA to ribosomal RNAs. Mean when they are present in a pre-form. So that will be converted into a mature form of ribosomal RNAs. Then if nucleotides are methylated, that methylation, mean adding of the methyl group, can be done with this SNO-RNAs. Then conversion of uridine to pseudo-uridine is also with the help of SNO-RNAs. Third type of RNAs are small cytoplasmic RNAs. They are found in the cytoplasm. The sedimentation coefficient is 7s. Now they can bind with the protein particle to form signal recognition particle. Now this signal recognition particle or SRP will help in binding of the ribosomes with the endoplasmic reticulum. We can see here, this is the endoplasmic reticulum, and these are the ribosomes binding to the endoplasmic reticulum, and this is the polypeptide chain. So this will help in formation of excretory proteins with the help of endoplasmic reticulum. So this is about catalytic RNAs, that is small nuclear RNAs, small nucleolar RNAs, small cytoplasmic RNAs. Now, translation is the process in which three of the RNAs they are helping: messenger RNA, ribosomal RNA, and transfer RNA. Now we can see in this figure. Now this is the messenger RNA. This is a smaller subunit of the ribosome. This is the bigger subunit. This part is transfer RNA. This is codon. This is anticodon, codon, anticodon. And this transfer RNA is carrying the amino acid. This is the formation of the polypeptide chain. So polypeptide chain will be later on released from the ribosome, which help in formation of the protein. So this whole process, translation, is done with the help of messenger RNA, ribosomal RNA, and transfer RNA. This is all about different kinds of RNA and their functions and what is RNA. If you like my video, please like, share, and subscribe.